Nobel Assembly at Karolinska Institutet, it is a great honor for me to introduce the 2017 Nobel laureates in physiology or medicine, Jeffrey Hall, Michael Rosbash, and Michael Young. They have received this year's Nobel Prize in physiology or medicine for their discoveries of molecular mechanisms controlling the circadian rhythm. Life on Earth is adapted to the rotation of our planet. For many years, we have known that living organisms, including humans, have an internal biological clock that helps them anticipate and adapt to the regular rhythm of the day. But how does this clock actually work? Jeff Hall, Michael Rosbash, and Michael Young were able to peek inside our biological clock and elucidate its inner workings. Their discoveries explain how animals and humans adapt their biological rhythm so that it's synchronized with the Earth's rotations. Using their beloved fruit fly as a model organism, this year's Nobel laureates isolated a gene that controls the normal biological rhythm. They showed that this gene encodes a protein that accumulates in the cell during the night and then is degraded during the day. Subsequently, they identified additional protein components of this machinery, exposing the mechanism governing the self-sustaining clockwork inside the cell. We now know that nearly all cells in our body have an inner clock, and to quote one of the laureates, Michael Young, we're just all a collection of clocks. We also know that a large proportion of our genes are regulated by the biological clock, and consequently, a careful, calibrated circadian rhythm adapts our physiology to the different phases of the day. The biological clock regulates critical functions such as behavior, hormone levels, sleep, body temperature, and metabolism. Our well-being is affected when there's a temporary mismatch between our external environment and this internal biological clock. For example, when we travel across several time zones and experience what's known as jet lag. We also know that chronic misalignment between our lifestyle and the rhythm dictated by our inner timekeeper is associated with increased risk for various diseases. Since their seminal discoveries, circadian biology has developed into a vast and highly dynamic research field for our health and our well-being. So in addition to this year's Nobel Prize, Jeff Hall, Michael Rosbash, and Michael Young have been awarded numerous prizes along their distinguished careers, including the Gruber Prize in Neuroscience, the Louisa Gross Horwitz Prize, the Shaw Prize, the Wiley Prize in Biomedical Sciences, and the Gardner Foundation International Award. As they say in the field of chronobiology, it is time to hear from each of the laureates. And I'm the timekeeper for this afternoon's session. We'll hear 3 individual lectures, and this really is a unique opportunity for us to gain first-hand insight into their journeys, and these journeys that have led to their seminal discoveries of mechanisms controlling the circadian rhythm. These journeys have likely been fueled by scientific inquiry, little mischief, hard work, teamwork, maybe a little friendly competition, and of course one cannot forget their love of genetics and that of the fruit fly. So now I'll begin with the introduction of the first of our 3 laureates, and this is Professor Emeritus Jeffrey Hall. He's a geneticist and a chronobiologist, and he is a Professor Emeritus of Biology at Brandeis University. Professor Hall received his doctorate degree in 1971 at the University of Washington in Seattle. He was a postdoc with Seymour Benzer, who was a pioneer in the field of genetics, and he performed this postdoc at the California Institute of Technology. He joined the faculty at Brandeis University in 1974, and in 2002, he became associated with the University of Maine. He was elected into the U.S. Academy of Sciences for his revolutionary work in the field of chronobiology. Professor Hall has spent his career examining the neurological components of fly courtship and behavioral rhythms. Now I'd like to ask you all to join me in welcoming this year's Nobel laureate, Professor Jeff Hall, up to the stage, and we will hear a lecture from him entitled The Little Flies, Multifaceted Basic Research Coming Out Better Than Intended. Professor Hall.
Do I just start talking?
You just start talking.
Okay. So, as Willie Loman would say, isn't this a remarkable thing? That alludes to the fact that I'm extremely grateful and deeply honored for all of what's going on here and this week. Beyond this week, whether or not the whole deal makes my mind boggle at the thought. Mind? But this leads me to segue into some claims that I'm going to make some additional remarks having to do with gratitude in certain general crypto-historical ways about various predecessors that ended up being at least loosely connected with the research in question, and further words of gratitude to certain specific biologists and geneticists. So, for example, not too long after I was attempting to establish research at dear old Brandeis University in the Northeast U.S., as a pleasant surprise to me, I was invited to give a presentation at the National Drosophila meetings. This had been kick-started by my second mentor in my time in the late 1950s, estimable geneticist named Larry Sandler, rest in peace. And many years later, I was invited to speak at the Drosophila meeting in the late 1970s or so, by which time the meeting had grown to be at least numerically more significant. And I was wondering, why did I receive such an invitation? I knew why. Because another mentor of mine, Dan Lindsley, a close colleague of Sandler, invited me as a favor. He was one of the meeting organizers. So I had just started attempting to establish a research enterprise, studying at the time, as you heard brief mention, fruit fly courtship behavior. Studied potentially studyable genetically, and therefore having the potential further to dig into the neural substrates of this kind of fruit fly behavior. So I was trying to fill out 30 minutes or so with something or other about our first blush studies of courtship. Really didn't have much to say. So a key part of this presentation had to do with a infamous courtship reproductive behavioral mutant, Drosophila, which have been rattling around the fly world for several years and nobody was actually studying it, which is perhaps for good reason. But it did lead to the creation of certain underground movies that showed a group of this genetic variant type in a wild frenzy of courtship-like interactions between and among the flies in this filthy film. So one of the members of the audience at this Drosophila meeting, sitting near the front, quipped, "This is like I Am Curious Yellow Forked." That's a joke. Not a funny joke, but it has the benefit of referring to one of my favorite movies that came out of this country, or maybe it was I Am Curious Blue. And the final feature of this quip, the English words yellow and forked, makes an overt reference to a certain type of genetic in Drosophila of the chromosomal type. Many genetic variants over the years, harking back to more than 100 years ago, were above the level of genic changes, instead chromosomal level changes, anomalies, with the designator that included, I say again, yellow forked. Okay, so those movies meant a whole hell of a lot to me, certainly more than various Bergman oeuvres, which included things like the protagonist playing checkers with death or something. I didn't understand any of that. But the fellow who made this tasteless jape from the audience was somebody who, in principle, should be known about by many in this room, including those involved in the prize awarding organization. Because the fellow who said this, who often behaved like he was as a dirty, dirty man, was Edward Lewis, who received this prize in the mid-1990s for his work that included a heavy dose of Drosophila genetics. And Ed Lewis was a student, back when he was a baby, he was a student of one of the giants in genetic history, one Alfred Sturtevant. Sturtevant was the academic offspring of Thomas Hunt Morgan, who was the first Drosophilist to be awarded this prize in the 1930s. And Morgan generated a variety of academic offspring, notably a famed trio of what you might call Morgan's F1 that included Sturtevant. And another member of this trio was another, at the time, fledgling Drosophila geneticist named Hermann Muller. Now, Muller, based on a discovery he made at the University of Texas in the middle, south of the United States, in an oasis of intellectual activity within that state, where, by the way, Mike Young, whom you, of course, hear from today, Mike Young did his PhD thesis work at the University of Texas. So Muller did work then which seemed to be very salient at the time, at least, and that led to Muller being awarded this prize in the 1940s. For Muller to accomplish what he did for his prize-winning study, it was quintessential for him to have designed and generated a particular kind of Drosophila chromosomal variant, which made all the difference for the performance of that study. This is a category of chromosomal variant, which I won't burden you with detail-wise, which became one of the great secret weapons of Drosophila research over many, many subsequent decades, without which huge number of Drosophilists could have never done anything if it hadn't been for this type of chromosomally based variant. Now, back to the matter of another type of chromosomal variant that I've already referred to that includes the designators yellow and forked, it was Muller himself who generated that now classic type of chromosomal variant in the 1940s. Although he didn't get the Nobel Prize for that. It was for other features of his genetically based studies. But these chromosomal variants, which were often designed deliberately and constructed via the usual cytogenetic mumbo-jumbo, were powerfully applicable to ask and answer some genetic questions, what I call genetic genetics, about hereditary factors. But the variants were maintained, survived down the generations, the fruit fly generations and the human ones. And several of them turned out to be massively applicable for other reasons later on. Okay. Now, back to Ed Lewis, and irrespective of what he said from the front row during that fruit fly meeting when I was rambling on about courtship behavior, here I need to mention another of the third member of Thomas Hunt Morgan's famous trio, his F1. This is Calvin Blackman Bridges, who made massive contributions in genetic history, including establishing major elements of what is known as fruit fly cytogenetics, observing and analyzing chromosomes, including the chromosomes contained in these aforementioned types of genetic variants. When my first mentor in my time was an undergraduate, yes, it was during the 30 Years' War, my first mentor was named, rest in peace, Philip Ives. And he was supervising a small coterie of undergraduates, and we were doing meager little fruit fly genetics projects. And he was a superb instructor, massively enthusiastic, motivational. He really taught us a whole hell of a lot of what to do on a given would-be research day. But also, he imbued his charges with what I realized later was described by the phrase, the lore of Drosophila, the deep background of research, initially genetics, involving this organism, which goes back more than 100 years from now. And Ives, like Ed Lewis, Ives was a student working under Sturtevant in the southwestern U.S. in what some would call, I never would call, the dumb blonde of the United States, near L.A. But in any event, one of Ives and Lewis's lab mates was Bridges. When Morgan moved from New York City, northeastern U.S., to the Los Angeles area, he brought, it was in the late 1920s, he brought with him Sturtevant and Bridges. Muller, as he grumbled about for the rest of his life, was exiled to places like Texas and Russia. But anyway, Ives told me when I was beginning to get a little bit of a feel about what fruit fly genetics can ask and answer, potentially, Ives mentioned that Bridges, who was his academic uncle, if you will, Bridges had the best eye to notice novel sports, you could say, variants at the anatomical level. He had the best eye to detect these as he and his lab mates would plow through these enormous mounds of fruit flies under the microscope, searching for rare, novel anatomical changes. And one of those that Bridges found back in New York in the 19-teens was immediately, as all the other sports, immediately rolled into their genetic processes where they asked, does this anatomical change have a genetic causation? And might it be hard specifiable that way? And many of these variants had that attribute. And so based on formal genetics, they started to create lots of information about the nature of this insect's genome with regard to genetic variants, including genic variants in addition to chromosomal level variants. But this particular genetic variant that Bridges, which is quite subtle, that Bridges detected in 1916, seemed as if it might be also biologically interesting, potentially, as opposed to just an extra genetic tool in the genetic variant toolkit. Biologically interesting. Compared, for instance, with some of these variants that had, for instance, altered eye color. Snore, not necessarily that interesting biologically. So Lewis, working under Sturtevant with Bridges as his lab mate, and with Ives as another lab mate, who divulged to me, part of the lore of Drosophila, about what a great eye Bridges himself had to notice novel variants. Lewis began his career after his PhD research studying purely genetic phenomena in Drosophila. But a while later, Lewis made a lateral move, just as a duck might make a lateral move à l'orange. I call part of my internal argument biogenetics. And he picked up on Bridges' variant, the one which seemed to Bridges and other of his lab mates to be interesting potentially at the biological level, more than just genetics itself. And Lewis, for several decades subsequently, pounded away at the factors pointed to by that Bridges variant and many additional related genetic variants, related genetically and biologically, and initiated the majority of Lewis's career in studying the animal's development by taking a genetic approach. When I was fortunate to get to know Ed well and realize that he was a developmental geneticist par excellence and a funky little fellow as well, cute as a button, by the way, Lewis was at Caltech toiling in absolute obscurity, even within that department. He was ignored or disparaged in the main. He was kind of a recluse. His members of his lab, I think, was one other person. But those who knew about his presence said on his behalf, well, Ed doesn't publish anything. This is now the early 70s. He had not published a paper since the mid-50s. They said he doesn't do that because he's a perfectionist. He demands absoluteness and perfection before he puts pencil to paper. And this got back to Ed, naturally, and he once said to me in private, he said, Jeff, this is caca. He said, I'm not publishing anything because I haven't found anything in living memory. And that was reality. But a few years later, in the late 70s, Ed burst forth with a publication, late 70s, that won him the Nobel Prize. And this was all based on the genetic variant at root found by Bridges back in the 19-teens. So this variant and many associated variants, including chromosome aberrations, mind you, of other categories, seem to say a whole hell of a lot about how certain genes help construct this organism beginning at the earliest stage of development. This paper, 78, also had strong evolutionary implications, even in the pre-molecular era. The relevant genetic factors in Drosophila or elsewhere had not yet been identified DNA-wise. Okay, so an example of how you can muddle along and admit to yourself that you're not getting anywhere, but you hope that you're making a tad of progress during a given half-decade. Keep going, and something might will out, as it did in the case of Ed's study, which is, thanks in part to his wherewithal, going back to Morgan, Sturtevant, with a lot of ancillary help from Muller and naturally Bridges, that this whole project that Ed was mired in when I first got to know him was an example of something coming out better than he intended, or at least expected. But as you also heard a mention of in the introductory remarks, another corner of the overall biogenetic enterprise, in principle, a priori, might involve Drosophila behavior, as opposed to developments, starting with the fertilized egg. Most conspicuously postnatal behavior after you get the mature adult animal. And so behavior biogenetics was pioneered principally, conspicuously, by the aforementioned Seymour Benzer, who by then had moved to Caltech and took advantage of Ed Lewis's local genetic and chromosomally based genetic expertise. So Benzer, instead of just imagining he might encounter the occasional behavioral variant, made a systematic attempt to generate behavioral variants from scratch and systematically search for them. The way he did this, the only way it would have worked was to apply, you guessed it, the aforementioned bibbledy-boo yellow-forked chromosomal variant, which made all the difference for this mutant hunt to work. And it did work. And several of these first blush behavioral mutants ended up pointing to well-definable genes with interesting products that function on behalf of the animal's postnatal activities, behavior, physiology, neurochemistry, as the case may be. But aping the Benzer approach, 1 year later, this is now the late, a student in Benzer's lab, Ronald J. Konopka, said, I'm going to do the same thing, the self-same genetic tactic, applying yellow forked, once again, very powerfully applicable, as came to be realized decades after Muller generated it. He said, I'm going to search for novel variants, but in this case of a particular type, daily rhythm variants that would show abnormalities or anomalies of daily biological rhythms in Drosophila. And what Konopka did, therefore, was kickstart, perhaps unwittingly, then a tiny sub-cottage industry enterprise. He kickstarted what is now, in this century, has become industrial-level research in chronogenetics and molecular genetic chronobiology, including neurobiology. The Konopka was a rare bird and very adept at what he was doing, both in terms of chronobiology and genetics, thanks to being mentored, as Ron was, by Lewis, largely. But he wasn't absolutely alone, even though chronogenetics was a tiny, tiny subset of the biogenetic enterprise. Because shortly after Konopka initiated the whole deal, some fungal geneticists did the same kind of thing. They searched for rhythm variants in a species of fungus, which should be appreciated by many in this room, because working on this fungal species, Neurospora, led to one of the banner Nobel Prizes in the history of the whole deal, when George Beadle and Edward Tatum realized that Neurospora genetics is ACEs, but they weren't studying genetic phenomena alone. They were using Neurospora genetics to ask questions about not just what is a gene. If you know what a gene is sitting there somewhere, that's not enough. What is a gene doing? What's coming out of a gene? And Beadle and Tatum made major contributions to what later became known as gene action, and which later on was often as a kind of a aphorism, DNA makes RNA makes protein. Okay, but Neurospora, at least in part because you can do fine genetics with that species of fungus, became also a chronogenetic system in that fungus after Konopka had initiated the fruit fly side of things. Neurospora chronogenetics, I have found, in my opinion, which is infallible, is ACEs. But at a minimum, I was very glad and very appreciative of becoming acquainted with the Neurospora fungal folk who are studying rhythms, because at the early rhythm conferences I went to, and you could never imagine the falderall at these conferences coming from the lecterns in the front of the room. It was bigger the imagination. And I would be sitting next to the Neurospora chronogeneticists in question, Jay Dunlap and Jennifer Laureates, and we would be sitting there rolling our eyes heavenward at what was coming out of a given speaker. And Jay or Jennifer would lean sideways and say, "Yeah, you're right. Forget about that. That is authentic chrono gibberish." But every once in a while, they'd lean sideways and say, "That's worth registering and processing later at the bar." So rhythm research, in part, when you get clear with various of the phenomena and the norms and the jargon and so forth, rhythms are interesting, I claim. Lots of research comes under the heading of dry as dust, often studied by Professor Dry as Dust. But rhythms are interesting in part because they're accessible to researchers. It just seems neat that biological cells and tissues can harbor clocks. Biological clocks, not electromechanical. Biological. What could they be? And back when I and Michael Rosbash and Mike Young got into this, nobody had a clue what a clock could be composed of or how it might operate. But we imagined that taking a genetic and eventually molecular genetic approach, the latter being quintessential, that we might contribute to understanding how rhythms are established and regulated, and how do they clunk along every day, all day. What am I out of time already? What? I'm out of my mind, which that's not possible. There is no mind left. But anyway, this brings me to another claim, which is cavalier and whimsical, is that I believe that this year, with regard to this prize in physiology or medicine, oddly named, that there should be 5 awardees. There should be 5 awardees this year in this category. And one of them should be the little flies, which have contributed. You can't do this, of course. And it also, there can't be 5 awardees that flouts the rules, or as Americans would say, flaunts the rules. Most of them being illiterate. But awardee number 4 should be the little flies, because as exemplified, they've created massive amounts of biological, in addition to genetic knowledge, going back more than 100 years of time. And the 5th awardee, silly as this may sound, should be the phenomenon, rhythmicity, because rhythms are arguably interesting. And you've already heard about why they should be regarded as interesting, and you're going to hear much more about that in meaningful words and pictures from Professors Rosbash and Young. But rhythms are interesting. And as you'll be hearing about, they're ubiquitous among organisms and even within a given type of organism. They're universal attributes, descriptively, seems to point to how important they must be, and as if they may have, in fact, been one of the earliest things that evolved, as Americans would say, 6,000 years ago, when life arose here on the third rock from the sun. But to study for mutants with abnormal rhythms in fruit flies and then Neurospora, and later a variety of other organisms, that those mutations had interesting abnormalities, but the mutations causing the mutant rhythm anomalies, the mutations also pointed to genes. And as Michael has said, even in private and even in mixed company, Jewish and Gentile, that for this biogenetic enterprise, chronogenetics, it was quintessential. You could never meaningfully predict what these genes would be about, what kinds of products would come out of them, unless the genes and the products were identified empirically by actual lab work. So the molecular side of the chronogenetic enterprise quintessentially needed to become and did do molecularly as well. So the final things I'll say here, having given a tip of the hat or so to various predecessors, including my two mentors and their associates who I wished I were alive when Bridges, Sturtevant, and even Muller, whom I never met. I begged my mentors to introduce me, and they refused. But many of my predecessors that I did or did not have anything to do with personally meant a hell of a lot to me. But springing forward into the 1980s and beyond, I think it's worth mentioning that there actually, of course, can only be 3 awardees in this category this year, as every year, 3 or fewer. And the 3 awardees are what, in terms of American research grant jargon, are PIs, the 3 PIs. That stands for principal investigator. Perhaps the adjective is correct, although not all PIs actually investigate, as you meant. But part of my own internal lingo was to refer to lab mates, such as in the lab of what I nominally supervised and Michael did and Mike did, and those two still do, are AIs, actual investigators. And I can't help give a tip of the hat to a subset of these, those who voted with their feet and their pocketbooks to travel long distances to be part of this week of bibbledy-boo, such as people I can never forget and never fail to appreciate, chrono-wise at least, like Bambos Kyriakou and Kathy Sawicki and Ralph Stanesky and Joel Levine, some of the major contributors to the chronogenetic, molecular, neurobehavioral. And then when Michael Rosbash and I, when half of each of our lab joined forces, one of the great joys, believe it or not, even I'm a terrible sourpuss, but I really genuinely enjoyed coming to what I laughingly called work, because I realized that there were all these fine AIs nominally under me, but also in Michael's lab. People, I know that some of them have made this trip for this week, like Paul Harden and Ravi Alada, but there was really a doubling or more than a doubling of AIs with whom to interact. And AIs in the 2 labs got to do, both in terms of vocational and personal matters. To have a large bi-lab research group, everybody studying in various different ways, an arguably interesting phenomenon. Very studyable in the little flies. And then later, and I'll never forget this, and I'll never fail to appreciate what she did. This is now vaunted Professor Leslie Vosshall, who was, when I first met her, was in the early 90s, was a baby working in Mike Young's lab in New York City. And at some dumb meeting, which are always dumb, scientific, but the one thing I remember from that meeting, Leslie Vosshall single-handedly kick-started a situation where the 3 labs, the 3 chronogenetic molecular chrono, were beginning to communicate. And this made the whole deal better professionally and personally. To get to know AIs in Mike's lab, such as Professor Vosshall and Amita Segal, who is here, who traveled a long way, and several other AIs. There are many, many other AIs among the 3 labs who I haven't mentioned because they didn't travel to Stockholm, or I'm unaware that they did or didn't. But it was just, I mean, you know, your supervisees make all the difference in terms if anything is going to happen. And it's often just on a day-to-day basis, interesting and enjoyable to interact with them, even if the PI is not really supervising. Like, Michael and I in one office or the other sitting down and talking about sports outcomes, agonies or something. But nonetheless, the AIs, between two labs and then with a third lab from the same region of the United States, but far away, it really became a three-lab group in terms of communication and even palpable cooperation. So the whole thing was, and I think it did come out better than we intended, at least at the beginning, going back to the late 70s. But however it was going to come out, it was really almost sensually enjoyable to deal with the little flies and all the potential that they create, including genetics, and then molecular and genomics. And they behave in a very complex, sophisticated way, more sophisticated than the behavior of many humans, which is like shooting fish in a barrel. But they divulge. It's almost like in some mystical, stupid way that you said, my God, if you seriously and if you know what you're doing and you care, I'm going to give up a secret or two. If you just keep going, even if for a while, if nobody's necessarily paying any attention, as in the case of the vaunted Ed Lewis, rest in peace, who was, as self-admittedly, he was always a very diffident fellow, was totally obscure even within his own department until he burst forth with studies which had spectacular heuristic value to understand not only Drosophila development, but developmental phenomena underpinned by genetic factors in all kinds of other animals. Michael's just waggling something at me as it wasn't quite an obscene gesture, but it should have been. But the same happened, again, better than we intended, that these fruit fly chronofactors, genic, mutants, mutations, molecular clones, connected with analogous and even literally homologous factors way beyond the insect arena, which I don't think we would have never particularly anticipated back in the late 70s. But Jesus, to work on the little flies and to be mentored in part about the lore of Drosophila, which didn't really help, it was just interesting and it was kind of motivational. So the tip of my hat to all these predecessors, all these successors, and especially, someday, come on, guys, give a prize to an invertebrate. It'll be funky, but warranted. So I'm done. Well, I'm really done in life as well as, but if there's been any attention, I thank you for it.
And so now we move forward. The clock ticks. Michael Rosbash is the Peter Gruber Professor of Neuroscience at Brandeis University, an investigator at the Howard Hughes Medical Institute. He's a geneticist, molecular biologist, and a chronobiologist. He received his doctoral degree in 1970 at the Massachusetts Institute of Technology in Cambridge under Sheldon Penman. Subsequently, he spent 3 years on a postdoctoral fellowship in genetics at the University. Since 1974, he's been on the faculty at Brandeis University at Waltham, where he's collaborated, as you heard, with Jeffrey Hall and investigated the genetic influences on circadian rhythms of the internal biological clock. Michael Rosbash is a member of the U.S. National Academy of Sciences. In more recent years, he has been working on the brain neuronal aspects of circadian rhythms. I would like to ask you all to join me in welcoming Professor Rosbash to the stage, where he will deliver his 2017 Nobel Prize lecture entitled The Circadian Clock, Transcriptional Feedback and the Regulation of Gene Expression. Welcome.
So it's a great pleasure to be here, a great honor. This whole business since the 2nd of October at 5:10 in the morning Boston time has left me virtually speechless. But being speechless is not really appropriate here, is it? So I'm afraid I'm going to have to screw up my courage and try and say something. So let me begin by trying to give you a little introduction to circadian rhythms. And first, as I like to do, to not forget this, I want to thank my current lab for the work they're currently doing, which I'll try and allude to at the end if I have a moment. Pay attention to the time here. And especially Kate Abruzzi, who's been with me for 16 years and has made the trip here to Stockholm to celebrate with all of us. And so circadian rhythms is a biochemical, as the slide says, physiological and behavioral adaptation to the rotation of the Earth. And there are two purposes to this. One is anticipation, namely the early bird gets the worm and the early worm avoids being eaten by the bird. And secondly, the clock manages to create internal coherence so that different reactions, different steps in metabolism, for example, happen in order and not all simultaneously. Oxidative and reduction reactions are happiest when they're separated in time. And so circadian rhythms arose a long time ago in response, of course, to this rotation of the Earth. And the rotation of the Earth was occurring well before nutrition was anything like what's currently available on our planet, and well before the atmosphere had its current gaseous constitution. And in fact, the oldest clock we know, the clock in cyanobacteria, really participated in oxygenation of the atmosphere. So 2 billion years ago, the planet experienced a reducing atmosphere, and it was really cyanobacteria with the aid of its clock that produced the oxygen that enabled life as we currently know it to arise. And so clocks exist really in different kingdoms, not only in bacteria, as I just alluded to, but also in plants and in animals, which I will focus on. And it's likely that these clocks arose independently because there's very little evidence of a relationship, especially between the cyanobacterial clock and animal clocks, and probably even the plant clocks arose independently. So this is such a powerful such a powerful contribution to fitness that it arose in these independent lines. And so a simple systems view of circadian clocks, a so-called skinogram named after Arnold Eskin, one of the early pioneers in the field, is illustrated here. And it simply demonstrates the fact that there are input signals, principally light, also temperature, which entrain the clock. The clock does not run exactly at 24 hours. Our endogenous period in humans, for example, is about 12 to 15 minutes long. And it's light every day, which resets our clock back to 24 hours. Those are the input signals. And then there are outputs, all kinds of things that we measure, we commonly measure, so-called the hands of the clock, which of course are not the clock itself. The clock are the gears inside, and the hands simply respond at various points to the clock itself. And it's really the nature of the pacemaker, the nature of the timekeeper, which is the focus of this lecture, in fact, the focus of this prize. And as Jeff alluded to, the beginning of this problem, certainly the beginning for us, not the beginning of all circadian work, but the modern beginning is really due to Konopka and Benzer, shown here on the bottom right, not at the beginning when Ron was a student in his lab, but much later in an interview. And these two people, and it was really Konopka who brought the circadian problem to Seymour. Konopka was interested as an undergraduate in circadian rhythms, and he suggested, as Jeff indicated, why don't we apply this genetic expertise to this biological problem in Drosophila. And they isolated 3 alleles of this gene they named period, and those 3 alleles either eliminated rhythms altogether. There was a short period variant and a long period variant, the so-called per 0 allele, per S for short, and per L for long. And remarkably, these alleles, these variants, had exactly the same effect on 2 completely different rhythmic phenomena, namely emergence of the adult from the pupal case, shown on your left, or locomotor activity rhythms, the running around of the fruit fly's activity rest cycle. Now we know their sleep-wake cycle, which were either absent, short, or long, exactly as one might have anticipated from the eclosion rhythms. So this was a magnificent contribution, and for those of you who chase high-profile papers, and perhaps this is a different angle on Jeff's Ed Lewis story, in the first decade after this paper was published, it had 10 citations. So the high-profile papers are not necessarily the ones which have staying power. And so at Brandeis, Jeff and my lab got together, and independently Mike Young's lab at Rockefeller, to clone this gene in the early days of recombinant DNA. And this is a picture of the group that did this: Will Zering, Tony James. The guy in the middle is Pranita Reddy. That's me, believe it or not, and Dave Wheeler. And so it was that group, and in fact, an additional group of people in my lab who I'm going to focus on. There were also people in Jeff's lab, but I want to sort of make sure that I acknowledge the people in my lab who contributed to this. Pranita I already mentioned, Chang Yu, Xin Lo, and Yoav Sitri, who unfortunately passed away. And that group really contributed to cloning and sequencing the period gene in my lab in the early mid-80s. And The outcome was that this was a pioneer protein. That meant this was such early days that the sequence identified no relatives in the database. The number of sequence proteins was so small that the chance of hitting upon a relative, a gene with a similar sequence, which might give you a clue as to what the protein was doing, just didn't exist. And so we spent several years struggling to try to find the promised land from the desert. And one clue for us was a paper that Chang Yu produced in 1987 that suggested that an intriguing repeat region within the protein, which turns out to be not very interesting anymore, was not necessary for circadian function. In other words, we could take the DNA that encoded the period protein, transform it into an arrhythmic animal, and we got perfectly good circadian function, which was missing this particular region. So that suggested that we needed to look in a different direction. And as science often has it, it turned out that the literature started to provide a bit of a clue for a direction to look at. And this paper was produced shortly after Chang's paper from Steve Cruz and Corey Goodman. And they cloned a Drosophila gene called single-minded. And it was a nuclear protein. They actually didn't know at the time it was a transcription factor, just a nuclear protein. And it had homology to the period protein. In fact, we were now no longer the pioneer. Single-minded was the new protein, and they benefited from having us there. Although, of course, since we didn't know what our protein did, it was of limited utility, except providing a high-profile connection for their paper. And so, nonetheless, the nuclear localization of single-minded suggested to us that gene expression in some way might be relevant to the function of the period protein. And shortly thereafter, Paul Harden, who was a postdoc in my lab, really produced what is arguably the most important paper from my lab, that the period gene product, the protein, was involved in a feedback loop which regulated the oscillations of its own mRNA. And this is a quantitation of those data. And the key thing, there are two key features of this. One is that the messenger RNA profile goes like this in time, and there are 24 hours between peaks. But if that experiment is done in a per short background in which the period protein has a 1 amino acid difference from the wild type protein, then the distance between those peaks is 19 or 20 hours rather than 24 hours, exactly mimicking the behavior. And so we proposed that this must be really important, perhaps central to timekeeping. But as I'll try and explain in just a couple of minutes, it was still a little bit of work to come up with a tighter, more restricted model. And this is a present that Paul gave me less than a month ago at a party we had, and this is the original gel showing oscillations, and the original paper under glass. It refers, it was a present that was framed, and so I cherish this gift. Thank you, Paul. And this was really not easy to do for two reasons. One is when you use whole flies, there were no oscillations that were visible. You had to use heads. That was his brainchild, because in the body there's lots of tissues which don't undergo oscillation, and that damped the signal, so it really wasn't visible. It's often in science a very small change which makes all the difference. And secondly, this is before PCR, so these assays were done by RNase protection, and that itself is a little bit of a story which I'll come to. So this particular gel gave rise to a model, and I call it a cautious model. This is the actual picture from the paper, the last figure of the paper. And what I want you to appreciate was that there was no mention of transcription in the title or the abstract, in other words, feedback loop. There was feedback loop, but no mention of transcription. And the way we imagined this was that it could work at the post-transcriptional level. For example, maybe messenger RNA half-life was affected, was regulated, and not transcription. It could be transcription. And moreover, it could be indirect. In other words, the period protein could affect these things directly, but perhaps the period protein affects another protein, which is actually the direct regulator, or most indirect, if you will, is that the period protein could influence behavior, which could in fact feedback and affect the molecular oscillations. In other words, behavior would actually be upstream as opposed to downstream of the molecular oscillations. So we were very conservative about this. And it turned out that over the next 2 or 3 years, that there were 4 papers that my lab produced, the first of which was really also Paul's work, which cemented the feedback loop model and emphasized transcription. So this first paper in PNAS, and an interesting aside is we really had trouble publishing this paper because everyone assumed this was transcription, and my lab worked on post-transcriptional regulation in yeast, half of my lab, and so I was well aware that this was not a slam dunk. This was not for sure to be transcriptional. And what this paper showed was the fact that the pre-messenger RNA, the intron-containing RNA, also underwent circadian oscillations, essentially indistinguishably from the messenger RNA, and also you could take the upstream region, the promoter region of the gene, and you could put it in front of a reporter gene, in this case choline acetyltransferase, CAT, and that regulatory, that regulatory DNA would oscillate. That reporter RNA would oscillate in exactly the same way as the per RNA. So all you needed was the upstream DNA, really indicating that this was very likely to be transcription. So the model strongly favored transcription, and then over the next couple of years, 3 more papers really cemented this model. One, our only collaboration with the aforementioned Seymour Benzer, in which immunoelectron microscopy was done to show that the period protein was really nuclear. If we constitutively overexpress the protein, this work done by Honghui Zheng, who is an eminent neuroscientist today, the scientific director of the Allen Institute, then this would clamp period mRNA levels very low and non-oscillating, consistent with a repressor function. And lastly, the region of homology between single-minded and period, and then a third protein, then so-called named the pass domain, turned out to be a dimerization domain, which could account for how period would work as a repressor, namely by associating with a positive transcription factor, which contained a pass domain. And this study was led by Josh Wong, who is also an eminent neuroscientist at Cold Spring Harbor today. So that really led us to the negative feedback loop model at the level of transcription. And I should say at this time, or shortly before this time, I won a bet from Mike Young, a dinner for the 4 of us, their wives, that gene expression would be the focus of the function of the protein, and he paid his debt, and we had a lovely dinner. And so mammals entered the story at this time when Joe Takahashi and his colleagues initiated and carried out a really heroic genetic screen in mice, in some ways encompassing the original Konopka and Benzer work, plus the cloning and the rescuing and the sequencing of the protein, and came up with a mutant which affected circadian rhythms very dramatically. Fortunately for them, the fifth mouse, which was several standard deviations off the norm for the period that they would normally see. Here's the wild type phenotype, and here's that particular mutant mouse. And of course, they named that mouse, they named that gene clock, and it turned out to be a Pas-containing transcription factor, which plays a role in this story. And at the same time, in my lab, Ravi Alada, who's here, and Joan Rudala were involved in a lengthy genetic screen to try to find other circadian mutants. And we knew about the remarkable work in the Young lab, and we didn't want to duplicate what they were doing, looking for period-altered mutations. So we actually looked at arrhythmic mutations, and it turns out that arrhythmic mutations are very easy to find. We had more than 40 of them, but they're very hard to sort out, to distinguish interesting ones from uninteresting ones. And so we devised a subscreen, and the subscreen for these mutants was to look at which of the strains had non-cycling and very low period and timeless levels. The idea being that if we had hit upon the positive transcription factor we were looking for, the X in our diagram, then there should be no cycling and low levels. And in fact, we only found three mutant strains which met that criterion, and 2 of those turned out to be alleles of cycle, or BMAL, the ortholog in mammals, and 1 of them turned out to be an allele of clock. So in fact, we had closed the circle, if you like, and managed to identify the positive transcription factor, the heterodimer that drives period and timeless, which then represses clock and cycle, and it turns out 3 of the same 4 proteins function in the same way in mammals. And so this is really conserved in the 550 million years or so of evolution since flies and mammals diverged, clearly present in the common ancestor that lived long ago. And of course, I'm focusing on transcriptional regulation, but there's a whole world of post-transcriptional regulation that Mike will allude to. For example, the function of timeless and double time genes that were isolated in his lab. So this is now a common theme, as you've already heard, among circadian oscillators, Neurospora plants, mice, where an activator gives rise to the synthesis of a repressor, which then increases in concentration, and then that repressor feeds back in the nucleus, interacts with the activator, turns off synthesis of the repressor, the proteins, the messenger RNAs and proteins decay away, and the whole system starts again then. So I just mentioned very briefly something contemporary just to show that this particular aspect of investigation is not over. This is a ChIP-seq profile for the active scientists who might be interested in the audience. This is a 6-time time course from the morning and then throughout the day to the evening. And this is the association of timeless or period with the upstream region of the DNA with the so-called E boxes. This is when the repressors associate. And the black arrows indicate the time at which clock, or the positive factor, associates with these same regions. And of course, not surprisingly, there's a delay between the association of the activator and the subsequent association of the repressors, so the repressors build up and then associate to turn off transcription. And it turns out to be quite interesting that the profile, if one does exactly the same kind of assay, but in this case actually tries to identify the proteins by mass spectrometry that are associated with the clock site complex on chromatin, that there's a much more discrete association of period and timeless at the time of maximum repression. In other words, by ChIP-seq, the proteins associate continuously, whereas by mass spec, there's a much more discrete feature of association. And so there's something that's very different that happens at a very particular time on chromatin that's associated with this repression step. And this is only to say a word about this particular kind of assay. And so beyond the feedback loop, I just will say a word about the fact that we and others have tried to address the notion of how much oscillation is there really. And in a paper that we published in 2001, we made two findings. One is that there are a large number of cycling RNAs in fly heads, much beyond this very constrained small feedback loop. And secondly, that oscillation, none of these cycling RNAs happen without a functional clock-cyc complex. So in other words, the small feedback loop really does sit at the top of a hierarchy, which gives rise to a large number of oscillating transcripts. And of course, the current view, the way to put this together and create a cartoon to rationalize this, if you like, is the fact that this small feedback loop here, which governs timing, there are a few more, a small or perhaps not so small number of direct target genes. Those direct target genes include other transcription factors, and those other transcription factors lead to the oscillation of secondary target genes and output functions. And it's really this idea, coupled with the fact that there are in mammals, flies as well, there really are clocks in all tissues of the body, not only the brain, but liver, kidney, the adrenal gland, pancreas, skin, lung, heart, etc. And it's really the fact that clocks are present everywhere. I think Uli Schibler gets the lion's share of the credit for really pointing out, doing a seminal experiment to indicate that this is the case. And the combination of the large number of cyclers indirectly produced from the feedback loop, plus the many tissues in which this occurs, gives rise to the fact that in the entire mammalian organism, more than 50% of all gene expression is under circadian control. So in other words, in excess of half of the 25,000 genes that we manufacture are governed by the circadian clock. And it's, of course, because of that feature that this picture makes really some sense, the fact that all kinds of phenomena that we're interested in, including sleep, which you'll hear about from Mike Young, and metabolism, and all sorts of other things, are really under the control of the circadian clock. So I'm going to just say, as it was present in the introduction, that my current interests are really these 75 neurons which function in the fly brain, and within which these red neurons, within which the same circadian feedback loop occurs in sync. And the question that we're quite focused on is, how do those neurons give rise to this very complicated behavioral profile that's characteristic of Drosophila and many other insects, namely a morning peak of activity, a siesta in the middle of the day. As Jeff indicated, these fruit flies are very smart. A peak of activity in the evening, and then sleep at night. And that whole profile is really controlled by those 75 neurons. And I've also become a pseudo neuroscientist, and so we're quite interested in how to assay firing patterns of discrete neurons in wake behaving flies. So flies that are running around and doing all kinds of behavior, how can you really assay the firing patterns of flies under those non-constrained conditions, and firing patterns of very discrete subsets of neurons. So I think I will turn in the last couple of minutes to a couple of philosophical points and try to address this question: why am I receiving this awesome prize, and what did I actually contribute? So that's, as Jeff told you about, it's the AIs which really do the work, and a PI who is ever so slightly introspective asks himself this question from time to time, and especially at times like this. So I am persistent, that's for sure. That is a characteristic of mine. I worked for many years on yeast post-transcriptional regulation, and that yeast lab, of course, I didn't have two labs. I had one lab with two themes, and the people were all intermingled. And so knowing about nucleic acids really helped our work, both experimentally, as I alluded to from this RNase protection experiment of Paul Hardin's, and also my training. The fact is that circadian rhythms had been dominated by neuroscience, and neuroscientists are interested in things that happen at the millisecond level or even sub-millisecond. But this didn't make any sense for phenomena which operates on a 24-hour time scale, but the half-lives of RNA and proteins does make sense. And it was with that knowledge that some of this work was done. And then, of course, I was smart enough to stay out of the way of talented people and let them make progress. So there's a lot of luck and good fortune that goes into work, that goes into life. My attaché this week, if you can believe it, is named Per. So I when he introduced himself, I said that I lead a charmed life. This could not possibly really happen to me. That is the nickname of the gene, of course, that we worked on. So more substantively, so I came to Brandeis and I encountered genetics. I was not a geneticist before I came to Brandeis, and Jeff was one feature, but only one of several people at Brandeis who were interested in genetics. Recombinant DNA came around just at the right time for our career. This was a perfect storm. I think for those of you who like movies, and Jeff introduced this metaphor, I could say that recombinant DNA was our Ingrid Bergman, and to quote from Casablanca, of all the gin joints and all the towns and all the world, she walked into ours. And you might not know, but the Epstein brothers, who wrote the screenplay for Casablanca, had two different endings. And on one of them, Rick Blaine gets in the plane to Lisbon along with Ingrid Bergman, and so we're in Stockholm. We had the happy ending to this story. And so I also, the feedback loop was correct in general. That was not for sure. Tons of cycling RNAs. Most of animal physiology is under our control. Who knew? Who knew that's how it would come out? I was fortunate to be funded by remarkable, far-sighted organizations, HHMI and NIH. My university is a wonderful meritocracy. Those funding agencies are meritocracies, and my environment was great. I have already mentioned the fact that it's really the AIs who do stuff. And of course, I had this long friendship and collaboration with Jeff Hall. This allowed us to put our two labs together, as he mentioned. People in his lab like Ralph Stanevsky, John Ewer, I got to associate with co-mentor, if you will, in the case of John. Patrick Emery in my lab, who's not here, collaborated with Ralph on the cloning of cryptochrome. So, and then of course, this had an element, a strong element of personal friendship. We have strong personality differences, Jeff and I should say, but we embrace those differences. I think it was really an asset to our work, and certainly enriched me personally. Ralph Greenspan likes to say that we made him think of Gilbert and Sullivan, who are arguably the most productive collaborating duo in music, and absolutely drove each other completely crazy in the course of their collaboration. So here's a list of the people I work with, and I want to particularly mention Ravi, Isaac, Patrick, Josh Wong, Shin, Pranitha. These are the early day people: Joan Rudula, Wei Ching, Chang, Hong Kui, and Larry Zweibel. They're the people who contributed to the early days of this story. I had a yeast lab, as I mentioned, and those people have gone on, many of them, to fame and fortune, and they were critical to my success. One of those yeast people became my wife, Nadja Abovich, and of course there's Nadja and I with our two daughters. And so as some friends of mine like to say, not only would I not be standing here without them, but I would be dead. And lastly, let me just say, and with family and Nadja as a segue, that science, my view of science, is a very romantic profession. What you're going to discover every day is an adventure, and it's really a great privilege, a joy, a romance, as I said. And I think I'll end on that note with a short poem, which is present in Ralph Greenspan's book, Fly Pushing, and it's written by his wife, Danny. And here's the poem: O genotype, O phenotype, this kiss had better last her. He's off to see his other love, Drosophila melanogaster. Thank you very much.
And so now we've come to what maybe we can call our third period in this afternoon. Michael W. Young is a professor at Rockefeller University. He is a biologist and a geneticist. He earned his undergraduate degree, as we heard, in biology from the University of Texas at Austin in 1971. After a summer of research with Burke Judd, and probably this emphasizes the importance of summer internships, he was working with the Drosophila genome, and that probably spurred on his interest in science because he remained at the University of Texas to complete a PhD in genetics in 1975. Michael Young continued his studies throughout his postdoctoral training at Stanford University School of Medicine with an interest in molecular genetics, where he worked in Dave Hogness's lab. From 1978, he's been on the faculty of Rockefeller University in New York. In 2004, Mike Young was appointed Vice President for Academic Affairs at Rockefeller University, and he was also granted the Richard and Jean Fisher Chair. Michael Young is a member of the U.S. National Academy of Sciences, and his research has shown how interactions among certain genes and their proteins produce molecular oscillations at the level of individual cells. I would like to ask all of you in joining me to welcome Mike Young up to the podium to deliver his Nobel lecture entitled Time Travels, a 40-year journey from Drosophilus clock mutants to human circadian disorders.
Bright lights. It's wonderful to be here. So I guess I'm the caboose. I'll pull us along and fill in a few gaps and tell you about some more recent work that's been on this journey. Let me start by just Jeff had talked about rhythms being all around us, and I think this is an example. My wife and I were out on a hike a few summers ago in New Mexico, and we came across this plant, which is called the Colorado 4 o'clock. And what I want you to notice is that all the blooms are closed, and notice this little branch here. I'm playing straight. This is at 2 o'clock when we're going out on the hike. 2 hours later, or a few hours later, we come back and we see this at 6 p.m. So this remarkable plant rhythm is believed to be due to the behavior of the pollinator, hawk moth, which is active at night in the evenings. And of course, you're in a desert environment. It's arid. Water is precious, and it takes water pumped into the petals of those leaves to open them up. So this appears to be a strategy, it seems, to conserve water in an arid environment and have those blooms available for pollination at times that make sense with regard to the behavior of your pollinator. Another example of these rhythms that are all around us is this record of hamster behavior that appeared in a symposium volume in 1960. This is each line is a 24-hour interval, so you're looking at 25 days in this animal's life in complete darkness the whole time. And it still amazes me when the animal gets on its running wheel, you get these deflections from that line, and you can predict within a couple of minutes each day when the animal's going to get on the running wheel and when it's going to get off. And you'll notice another feature of this is it's not 24 hours; it's about 24 hours. It's circadian, which, how could it be anything other than an internal reflection of some kind of an internal pacemaker. As Michael pointed out, we have still other phenotypes as we acquire new tools to look more deeply for rhythmicity. These are rhythms that are established in the head of the fruit fly. This was of Drosophila. This was work of Adam Claridge Chang and Hermann Muller about 15, 16 years ago. Using microarray data. And the way you read this is that this is 150 gene patterns stacked one on top of the other. Each row of red and green boxes is the pattern of a single gene followed over 6 24-hour intervals, some with a light-dark cycle, but others in complete darkness. And they're stacked according to time of day of their maximum expression. So these are maximally at dawn, at noon, these at dusk, these at midnight, and so forth. And I think what I hope you see are red and green stripes running top to bottom. just illustrating these waves of expression over this very large group of genes. And the other thing I'd like to point out is that those are not vertical stripes. They're running diagonally across the chart, which is pointing out for us that this timer is not simply swinging back and forth between two states, day and night. It's a program that generates patterns of gene expression where every hour of the day there's a unique relationship between genes that are hitting their peaks and genes that are hitting their lows. The next slide shows that same pattern, but now next to it are results that are obtained with 3 clock mutants, period, timeless, and clock. So those are null mutants, and where are the stripes? The stripes are now gone. And as Michael pointed out in his talk a few minutes ago, what we appreciate is that not only does the behavior go away with these mutants, but all of the genetic changes that are programmed, this temporal program is eliminated by loss of those genes. And this is unique to the genes that contribute to this pacemaker mechanism that we've been studying. So just to give you an overview once again of the way this pacemaker works, we have the period and timeless genes on different chromosomes in the nucleus. Transcripts are produced by association of clock and cycle, activating transcription of both per and timeless. Their RNAs are translated in the cytoplasm to give timeless and period proteins, but then we run into some post-transcriptional breaks on the system that produce the oscillations that we see. One of the important initial breaks is produced by a gene we called double time. This turned out, when cloned and sequenced, to be casein kinase 1. And that kinase phosphorylates the period protein and marks it for destruction. So at the early stage of each daily cycle, when period proteins and timeless proteins are at very low levels, double time, which is constitutively produced at high levels at all times of day and night, is the first partner that the period protein sees. And so most of the early period proteins that are produced are immediately dispensed with in the cell. On the other hand, many hours later, as timeless proteins build up, as we have higher and higher levels of timeless proteins, we now can produce something new, which is a period timeless dimer. And period timeless dimers are immune to this phosphorylation-based destruction by double time. And they also have a property that they now seek the nucleus, whereas period and timeless on their own seek the cytoplasm. And of course, as you've already heard, period and timeless are inhibitors. They bind to physically clock and cycle and repress the action of those activators to close this cycle. In a light-dark cycle, that would all happen in constant darkness, which is why we see oscillations principally in constant darkness. But in day-night cycles, we also have the issue of these clocks following the phase of the environmental cycle, and that's achieved in the fly by a protein called cryptochrome, which is a photolyase-like molecule that absorbs light. When it absorbs light, it physically associates with timeless, recruits proteins that will cause both timeless and cryptochrome to become phosphorylated and degraded, so that all day long, when the sun is up, we have no timeless because of the activation of cryptochrome. And of course, if you have no timeless, all the period protein will only have one partner, which is double time, which means we also have no period. So we have massive amounts of RNA for both period and timeless during the daytime, but it's sterile in the sense that we're not accumulating protein until the sun goes down. And this is essentially how the phase of this clock is established. And as we move a fly from New York to Stockholm, for example, you can see the effects that would be expected from this kind of a mechanism. So as Michael pointed out, there's a great deal of homology when we move from the fly to other animals, the rest of the animal kingdom. So in us, for example, we have many familiar proteins. We have clock and BMAL, which are the orthologs of clock and cycle in the fly. We have 3 period proteins, PER1, PER2, and PER3, all of which are related to the single period protein that we have in the fly. We also, once again, have casein kinase 1, the double-time ortholog in mammals, which once again controls the accumulation and fate of the period protein. One difference, an important difference we have is whereas cryptochrome was a partner for timeless controlling its accumulation, now cryptochrome has become the partner for PER, which is an interesting evolutionary switch in dance partners. But it's a cryptochrome PER dimer instead of a timeless PER dimer that now moves to nuclei and inhibits the action of clock and BMAL. And I'll come back to these differences in a few moments when I begin to speak more about human clocks. So originally, the discovery that clocks were found all over the body was a discovery in flies. It was made in Drosophila by Jeff Hall in collaboration with Steve Kay in 1997. And they had generated a form of PER which was connected to luciferase so that you could use a light monitoring scheme to image live Drosophila and all the intact and dissected body parts of the fly, and realized that there were PER-driven cycles all over the fly. And as Michael pointed out, we now know that much the same is true for mammals, including ourselves. And in this example, I'm just giving you examples of clocks that are found in the brain versus the lung, the liver, and skeletal muscle. And I want to suggest some of the things that might happen to you if you move from New York to Stockholm, or even when you impose a brain-body conflict experimentally in the lab that can be studied continuously for many, many days. So if this mouse is kept on New York City time, it's a nocturnal animal. It's going to be active at night. It's going to be sleeping during the day. If unperturbed, it will want to eat at night when it's active and be quiescent during the day. And we have these clocks in different tissues all agreeing on the time zone. They're all agreeing that they're working together on New York City time. Many years ago, Michael Menneker at the University of Virginia did an experiment in which he established a brain-body conflict by allowing the mouse to eat only in the middle of the day when it should be in the middle of its sleep interval. Provide food for an hour or so. After a few days, the animal learns to predict, will wake up spontaneously before the food is delivered, eat the food, go back to sleep, but then get on its running wheel and run on the wheel all night, as it would appropriately for New York City. What was found after a few days of this, though, is that the clocks, everywhere except the brain, had changed their phases by this new routine. So the brain is still stuck in New York. It's seeing that light-dark cycle. It's producing those activity rhythms that are appropriate at night. But the rest of the body is in disagreement, is not pulled along by what the brain thinks is going on. They're getting their cues directly from the feeding schedule or from other organs in the animal that are responding directly from the feeding schedule. So we have, in effect, an animal whose a collection of clocks that can't agree with one another. And this is remarkably similar to what happens when we undergo jet lag. Different organs proceed to the new time zone at different paces. We're very much this is very much a problem of a multiplicity of clocks that can't agree for several days when we change time zones dramatically in this fashion. What we do know from these kinds of experiments is if the animal is maintained for many days or even weeks, we begin to see a number of anomalous events in metabolism and behavior that are consistent with much more severe effects that come from actually eliminating clocks in these different subsets of organs. So let me turn to these that we've been doing in humans that consider a related problem, which is what if your clock doesn't agree with the environmental cycle? We've got a 24-hour environmental cycle. What if you're genetically programmed to have a clock, as we've heard in flies, like a per short or a per long? What happens if we have a clock that's out of sync with the turning of the Earth? And I'm going to be talking about a syndrome called delayed sleep phase disorder. We can think of individuals that have this disorder as night owls, extreme night owls. So they might be hammering away on their keyboards late at night, getting a big burst of energy off of their computer screen, which is, if anything, going to make things worse for them late into the night. And these are some of the attributes of these individuals. We think this appears to affect about 5% of the population from clinical studies. It's a persistent delay in the timing of the major sleep episode and strong resistance to efforts to try to manipulate sleep phase, to advance it, to make it easier to show up for work at 8 or 9 o'clock in the morning, for example. So these are records from two individuals that were in a study we began a number of years ago. On the left is a control subject, which we'll call Tau 18. And these are activity records, wake-sleep records, that involve two kinds of monitoring. One is with a activity monitor, and the other is just with entries in a sleep log that's kept by the bed, so that each day or evening when the individual gets into bed, they mark the time down. And when they get out of bed, they put another mark in the log. So we have two correlated pieces of data to consider. So the way the data are plotted here is to plot not just 24 hours, but 48-hour intervals in this process. And what this allows us to do is compare the time that the individual is getting into bed with the time that the individual is getting out of bed. So you notice in the control subject from both the sleep log and the activity graphs that there's a very neat and consistent pattern in which at about 11:30 to midnight this individual is getting in bed each night and rapidly thereafter going to sleep. And at 7 or 8 each morning, again, pretty consistently, they become active and they put a mark in the sleep log that they're getting out of bed. The DSPD subject, Tau 11, has quite an erratic record by comparison, but I think the most important features are if we look at the in-bed times versus the out-of-bed times, we see that this is an individual that's always getting in bed well after midnight. And the out-of-bed times, they're becoming active again, usually just a little bit before noon. So this is a striking phase variation with regard to the timing of their wake-sleep cycle. So we were able to study that individual and several members of the family, and did a whole exome sequencing of all of these members of the extended family. And we found that there were a number of those indicated in black or gray that had an aberrant pattern of sleep, and one cousin over there with a normal sleep pattern. And then the difference that came up by whole exome sequencing in these individuals gave us one most likely candidate, which is a variation at the gene cryptochrome 1. This mutation is a single nucleotide change. It's at a spliced donor site just downstream from exon 11. And that change predicts that exon 12 will be spliced now directly to exon 10, skipping the information in exon 11. So that would predict a 72 bases missing in the messenger RNA and 24 amino acids missing from the protein. And here are we had collected dermal fibroblasts from each of the subjects in this study. There were 20 or so subjects that we studied, and you'll see subject 11 is uniquely producing 2 forms of the RNA from cryptochrome 1 and 2 forms of protein in the skin, in the dermal fibroblasts. So clearly a significant change in the structure of both the RNA and the protein in this individual. We went on to ask whether or not that modified gene was enough to change the circadian clock of a naive cultured cell. And Alina Patke in our lab did this by starting with a cultured cell that had no copy of cryptochrome. Usually would have 2 copies, cryptochrome 1 and cryptochrome 2. If you have no copies, you'll be arrhythmic. The cells will be arrhythmic. And she used this as a test system to ask, is there a difference in the pattern of circadian rhythmicity at the cellular level given a wild-type form of cryptochrome 1 or this variant form of cryptochrome 1? And as you see here, there was a very consistent pattern of rhythmicity that comes from the wild-type form, the full-length form of cryptochrome, and this truncated form gave a pattern with a longer circadian rhythm, about half an hour longer than that provided by the wild-type gene. So clearly this gene is capable of producing a change in the circadian rhythm of the recipient cells. So once again, the difference between the fly clock over here and the human clock shown in the right, important new role for cryptochrome. So this of course is the gene that is now modified in this case. Cryptochrome, the work it does is in conjunction with PER to form that inhibitory complex that suppresses the activity of clock and cycle. So keeping that in mind, Alina took a look at the interactions among these proteins. We know from that model, from what's known about the human clock and the fly clock, that period and cryptochrome will be partners to form an inhibitory complex that will then further physically interact with clock and BMAL. And so she asked the following question: does it matter with regard to the physical association among these proteins whether you're the full-length form of this protein or the truncated form of the protein? So if I start down here, for example, and we have input proteins where we've created only the full-length protein is provided, or the truncated form of the protein is provided, or a synthetic heterozygote in which both forms of the protein are produced. What happens if you now do an immunoprecipitation with a target of hemagglutinin-associated clock? And what is found is that the full-length form of cryptochrome is very weakly recovered, much stronger recovery of the truncated form, and in this synthetic heterozygote, again, a strong bias toward the truncated form of the protein. Up here on the left is just a reciprocal experiment where the focus of immunoprecipitation is BMAL, and again the inputs are over here, and we see much the same response. The full-length protein is recovered poorly relative to the robust recovery of the truncated protein, and the synthetic heterozygote is strongly biased toward the truncated form of the protein. In contrast, if we look at what's happening with interactions with PER itself, how do these 2 varieties of cryptochrome interact with PER itself? We find, here's the input, that the input and the pull downs are the same. So in summary, what seems to be the case is that period and cryptochrome appear to have a strong physical interaction that is not affected by this mutation, but the interaction of the period cryptochrome heterodimer with its target activator proteins, clock and BMAL, is strongly altered by this mutation. So we thought all of this was extremely interesting at the level of molecular biology, but we were really stunned when we sat down at a computer a few years ago and asked, well, how common is this mutation in human populations? And this is what we saw when we did that, looking at the exact database available through Cambridge. The overall world population, the frequency of this allele suggests that carriers, and again, this is behaving as a dominant so far as we've seen so far, carriers would be almost 1% level in the population. If we focus on Europeans, those that are European or of European descent, the allele frequency is shown here, so twice that would be the carrier frequency. That's about 1 in 75 of European descent. So we were stunned that this allele, which had these features changing the function of the clock, was so prevalent, but it did suggest that we should be able to identify without a lot of trouble additional carriers to confirm this relationship. And we set up a collaboration with Tayfun Özalek at Bilkent University in Turkey, who had been working with a colleague across the hall, Jeff Friedman. And Tayfun recognized a number of families that had carriers, both homozygous and heterozygous carriers. Large families, only a few of which are shown here. And by looking at the patterns of sleep in all of these individuals, we're able to compare the patterns shown in the filled boxes, the dark boxes, carrier boxes, versus those in the unfilled boxes, which are non-carriers. And we can plot the behaviors of these two groups as shown here, the non-carriers, the shown over here, that have what we call a mid-sleep measurement on free days. So on free days, when do you go to sleep? When do you wake up? And what's the midpoint of that interval? And for most of us, that's going to be around 4:30 in the morning. And that's true for most of the non-carriers in these families, but their relatives who are carriers, whether heterozygous or homozygous, have a very different pattern that's shifted by about 2 hours, very much like the original proband that we studied, where the mid-sleep point is about the time that most of us would be getting up and taking off for work. There's a second phenotype that emerged, which are fragmented sleepers. These are individuals that point to their major sleep episode as a short nap that can be early or late in the evening. Again, something not seen in the non-carriers. So to summarize what we've seen, we've got a number of unrelated families in which the presence of this variation predicts DSPD, the high frequency, the penetrance, the high penetrance of this mutation with regard to that behavior suggests a significant contribution to DSPD worldwide. Since the mutation is found in multiple members of each family, it's running through the family, it's being inherited, we have to assume that every tissue has these altered clocks. The variation shows enhanced binding to clock and BMAL, as I pointed out, and it appears to be a strengthened transcriptional inhibitor by other work that I don't have time to talk about today. The competitive binding to clock and BMAL suggests a molecular basis for the dominance of this trait. And finally, as I mentioned, it's sufficient to express just this one gene in individual cells in culture and recapitulate an altered circadian rhythm. So lastly, I'd just like to point at some questions we have for the future. One of the problems in studying sleep disorders has been relationships to other medical conditions. It's long been the case that anecdotally a variety and psychiatric disorders are often said to be associated with disordered sleep and very often delayed sleep. And but it's been difficult or impossible to establish causal relationships. But what we have here, I believe, is a new opportunity. If you have large numbers of subjects that are available to study, we should be able to test whether a particular sleep mutation extends to other medical problems, and particularly when the mutation can be studied as it's traveling through unrelated families, we should be able to rule out the impact of environment and make some clear conclusions about the relationship of mutations of this sort to other medical disorders. And lastly, let me just point to the work, to the people, the AIs, as Jeff put it, in the lab that have contributed to this work over the many years. In the 1980s, Ted Bargello and Rob Jackson were involved in the initial mapping and cloning of the period gene and sequencing, identifying the nature of the mutations that Ron Konopka and Seymour Benzer had initially found some years before. In the 1990s, Leslie, Amita, Jeff, Lino, and Adrian all worked on the discovery and characterization of the timeless gene and its protein. Shortly thereafter, Jeff Price, Lino, Adrian, Justin, and Brian all worked on the discovery and characterization of double time, whose role I told you about. And in the 2000s, we began to move into the era of microarrays and looking at the larger patterns of gene expression that are controlled by these clocks, and that was principally driven by Adam Claridge-Chang and Herman Wienen. But still more genes to be discovered. Sebastian Martinek found a role for GSK3, which is particularly interesting still because of its sensitivity to commonly used drug treatments such as lithium. In the 2010s, or the last 7 years, Dragan Ragulja and Nick Stavropoulos had both initiated studies of a different kind of genetics, that of control of sleep duration, the homeostatic control of sleep, which, along with circadian regulation, ultimately controls the pattern. And as I mentioned, Alina Patka has been involved in every aspect of our human work. And I'll just show one last slide. I want to point first to Lino Saez, who was a member of the laboratory for almost 30 years. He was involved in almost everything that I spoke about today. Lino unfortunately passed away in 2014, but I know he would really love to have been able to be here. Alina Patke, again, has been responsible for every aspect of the human work. She's had her hands on every part of that project, and she is here, and I just want to thank her personally. So thanks for your attention, and for the honor.